Welcome to the Science and Beers podcast with me, Mick Mickey. Talking science and drinking beers with researchers down at the pub. We cover a new topic each episode, so join us with a brew and let's cheers to science. In February 1953, Francis Crick burst through the doors of the pub called the Eagle in Cambridge and proclaimed that he and James Watson had found the secret of life. They had, of course, discovered the helix shape of deoxyribose nucleic acid, or DNA. Our guest this week, Dr. Philip Hallenboer, is an expert on DNA. He has given a Science and Beers talk before on the subject. Philip has his PhD in molecular biology from the University of Southern Denmark, where he has also worked as a scientist in the Department of Biochemistry and Molecular Biology. He has recently swapped that role to act as a research advisor to help other scientists secure funding. This podcast was recorded in July during our summer break, and Philip and myself, we went to a bar in Odense, Denmark, Christian Fietel, and it was a typical beautiful Danish summer's day with clear blue skies one minute and torrential downpours the next. So you will be able to hear some other people in the pub. You'll also be able to hear the patter of rain on the parasol above us. But you'll also get to learn a lot about DNA, evolution, gene editing, and Philip's career paths. Not only is Philip a researcher, but he is a novelist, an author. He has 2 books published on the subject of science, and we'll get on to that later on. Hope you enjoyed this episode. Please consider supporting the podcast at patreon.com/Science and Beers. Thanks.
Yeah, thanks for having me once again.
Thanks for joining this podcast.
Be more than happy to.
Fantastic. It was a fantastic talk you gave at the first season.
Yeah, thank you very much.
Beers.
Yeah,
all them years ago.
Yeah. No, but I still remember it was a lot of fun.
Yeah.
So I've been to some of these talks for this Forskningsdøgn.
Yeah.
That's also been fun. But I think the Science and Beer is something unique, and the mood is something different.
Well, that's where the alcohol comes in.
Yeah, exactly.
Just add a couple of beers and you're all good.
Yeah. It always helps.
So let's take it from the top, Philip.
Yeah.
From the bottom up, what is the simplest component of DNA, and then how is that expressed?
So the simplest component of the DNA is a nucleotide. So that's a chemical component that holds information about that. So the assembly of these chemical components. So you have 4 different chemical components, 4 different letters in the DNA.
4 different nucleotides.
4 different nucleotides in the alphabet, on the DNA alphabet. A, C, G, and T, they are called. So that's 4 different chemicals, so to say, that are put together in this DNA string.
So the elements in the nucleotides are just slightly different from each other.
Yeah. So, but yeah. So there and what you when you put these together, that's the sequence on how these nucleotides, these elements, they follow each other. That's really the important. That's how you store information. So the sequence of these constituents, that's how you store information in the DNA alphabet or the
A, C, T, G, 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 A, C, T, A.
Yeah, the DNA code. So this whole string of these elements, that's you put this into these long strands of DNA. So you also sometimes call these chromosomes. And then these certain regions of this DNA, long DNA strands, that's where you have these genes and genes, that's where you translate this DNA sequence into proteins. So what the cell does is that it goes in and read these strings of A, G, and C, Gs, these elements, and then translate them into a certain amino acid that it puts into the protein. So upon translation of a gene, you assemble a protein based on the DNA sequence. So what the cell does is that it reads 3 of these nucleotides, and that determines a certain amino acids. Then the next 3 letters in the DNA alphabet, that determines the next amino acid that goes into this protein. And then you have, at one point when you have enough of these DNA letters or amino acid codes, then you have what's called a stop codon, and that tells the cell to stop making the protein here, and then you have this final strand of amino acids. And what happens is that these amino acids fold in a 3D structure and make a protein that can now perform a certain function in the cell. So going from nucleotides that are assembled one by one into all this long DNA strand, that the cell can then translate into this protein. That's how the cell sort of works and maintains its function.
Okay. And can we go over just how DNA is stored in a cell?
Yeah. So in
So you have your DNA, you have your genes.
Yeah. So it depends on which type of cell. So for a human cell, you have all your DNA stored in a specific compartment or organelle compartment within the cell. That's called the nucleus. So in the very core, you have this DNA stored, and there's all these. So we have 46 strands of DNA, 46 chromosomes that is mingled in between each other. It's actually it's kind of amazing that within each cell in your body you have 2 meters of DNA. So and given that you have 37 trillion cells in your body, so if you put all your DNA, it's sidetracking a little bit, but if you put all your DNA after one another, you actually within one human body you actually have something that corresponds to 2 times the diameter of the solar system. So you really, it's quite. So this very thin thread of DNA can actually span the solar system twice.
Okay.
That's really cool.
That's
Yeah.
That's a long book.
Yeah, that's
long book of information right there.
It's amazing that we have that. So, but of course, it's folded up together, all this DNA, and then the cell can go in, transcribe the DNA, and then start making the protein. And depending on which species you are, you store it a little bit differently. So for human beings, we store them as linear strands. Bacteria and so on, they have them as circular DNA, and they also have small pieces of DNA that they can actually send back and forth to one another and thereby share information. These are called plasmids. So there are different ways depending on which species you are on how you store them. So the problem with our way of storing it is that when you want to make a new cell, and you have to replicate the DNA, so you have to make an extra copy of the DNA. And when you have linear DNA, the way that it is done is that you always lose a little bit in the end of these long DNA strands. So that's why when at the beginning of embryonic genesis, and you have the DNA, the cell actually extends your chromosomes. It puts a lot of extra information, useless information in the end, called telomeres. So you extend the DNA with useless information, and this is then slowly chopped as you grow older.
Every time the cells divide, it's chopped.
Yeah, you use a little bit of this useless information, and then when sort of you get to where you have stored information, this cell can no longer divide, because then you start losing important protein recipes.
And is that death?
Yeah, so that's a way of aging, and so that's also why, for instance, a cancer cell, which can divide indefinitely, what it actually does, it reactivates this enzyme that add these telomeres in the end. So normally, when in our body, this on most of our cells, this enzyme that adds these extra extensions to the RNA, or sorry, DNA, that is no longer active. But cancer cells, they can reactivate that, and then they can just keep on dividing.
Well, hang on a second. You know, my question here is there a way to like take this part of the cancer cells to extend our telomeres?
So that's of course one way of addressing cancer. That is to
Well, I meant
to live forever, or
exactly.
yeah.
If you just
No, but of course people are looking into it, and some people are trying to do this, called biohackers, where they sort of reactivate this enzyme.
Yeah.
But I think there's a thin line between cancer and living forever, right? Because so cancer, that's uncontrolled growth, and living forever, that's controlled growth, you can say.
Yeah.
And we are definitely not there yet where we can control that.
Yeah.
So some years ago, there was scientists have developed a theory or a method where you can actually take a cell in your body and turn it into a stem cell. The problem is that you had to activate genes that are cancerous. So in order so the perspective, of course, that you can use any type of cell in your body and make a stem cell and then replenish whatever organ or whatever you have lost. But the thing is that these cells that you make in the culture, making them, functional cells and making them cancer cells, there's a very thin balance. Mm-hmm. So that's what people are struggling with at the moment.
So, dear listener, if you want to get into that area of research, please go ahead, extend our telomeres without any bad consequences. Control growth. Thank you very much. Can we talk a little bit about the discovery of DNA? If you know a little bit about that.
Yeah. So how it was, the structure and so on.
Yeah. Well. First of all, I would say going back already to Darwin.
Yeah.
So Darwin thought that there's inheritable traits.
Yeah.
In species, and then you had this guy Gregor Mendel, who noticed in pea plants.
Yeah.
Some are tall, some are short.
Yeah.
If you cross the tall with tall, some of them are tall, but maybe 1/4 are short as well. And he thought there must be something inside the cells of these plants that is dominant and recessive, and there must be two of them.
Exactly.
Whatever they are.
Yeah.
And whenever they combine, the dominant one is always expressed. But if there's two recessive ones, then that's expressed, and that led to the short peas.
Yeah. Yes. Yeah. That was all his laws on the genetics and how traits are inherited.
So I find that an interesting story in that okay, there's a bigger picture. There's something inheritable. Okay, there's something in the cells, and then you keep going, and then you're looking into what is DNA. Do you know anything about the discovery there?
So basically, how they showed that the DNA was carrying the traits, that they could see that they could, if they transferred DNA from one organism to another, they got this new trait that was incorporated in this DNA. So that's why how they discovered that the information is stored in the DNA, but the function is actually done by the proteins. So, yeah.
Yeah. How did they? So, I get where you're coming from. You're talking about like gene editing in cells. Yeah.
Yeah. Yeah.
Okay.
And gene editing, that sort of moving DNA around between different organisms, or modulating or modifying in a certain organism. But when you make what's called a GMO, a genetically modified organism, you transfer DNA from one organism to the other, and that was how they originally discovered that DNA carries information. I don't recall the exact setup of the experiment, but it was simply that they either transferred proteins to a similar organism or DNA, and then monitored which one got the new trait. So the proteins, they have a half-life so they die, and DNA that can be replicated. So you can still the new cell that got this new DNA will still if got the DNA, it will make it's capable of making new DNA and then new proteins. But if you only get the proteins, then the information will be lost because the proteins have a tendency to die out. So people also like to think of this as DNA as a cookbook or recipe book, whereas the proteins themselves are the actual dish that is made from these recipes.
So I'm picturing here now. You're talking about gene splicing editing, like a mouse, a fluorescent mouse with an ear growing out its back.
Yeah,
that's a strange image for anybody who's never saw that picture.
Yeah. No, but that's doable today to make.
It's doable to make a fluorescent mouse with an ear growing out its back.
Yeah. So
why would it?
So people wouldn't just be making these for fun. Of course, it has to make some sense or make some sense.
Yeah.
So the fluorescence comes from specific proteins, or one specific protein that you can express in these mice. It comes from jellyfish, and that can be used to track cells within the body or wherever you want to track cells or monitor how proteins move within the cells. So normally proteins, they move around in the cells depending on where their function is needed, and you can track their motion by putting on this green tag, and then study them in the microscope and to see how the cells move, because where the protein sits in the cell is really important for its functions. And the ear on the back, so that's sort of for a different thing. That's if you want to make if you lost an ear, for example, you can grow this. Sort of grow it in a dish. So that's where people are really interested in stem cells at the moment. Because what you do is that you make a 3D structure of an ear, and then you put cells from a given patient, stem cells from a given patient, onto this 3D structure, and then sort of incorporates all sort of whatever holes there are in this porous structure, 3D structure. And then you can, at least theoretically, transplant it back on the patient, and then they should have not a functional ear, but at least something that looks like an ear. So
well, that's a beautiful story. There's me talking about how it just the madness of making a fluorescent mouse with an ear on its back, and you've just given me there were actual intentions, scientific intentions that would make the world a better place with a creation such as that.
I think, or at least most all, it's fair to say that all scientists have this motivation that they want them to make the world a better place. Indeed. I haven't met anyone that's not interested in that.
The unfortunate thing is that picture got a few headliners with a few questions about why would they do that?
Yeah.
Frankenstein.
Yeah. No, but there are some. Of course, if you just look at this weird mouse, that it doesn't make sense. But there's of course an idea to it.
But it's amazing that we've learned that DNA is the code that makes all biology, whereas in modern times, the past few decades, we're able to read that book, read the instructions, and then take little bits that we need to improve things, to improve instruments, or to improve our health, and then take it out and put it into something else.
Yeah. A very popular tool these days is called CRISPR. So we go in and manipulate one single letter in this DNA alphabet or code. So and that is used every day in labs around the world to manipulate DNA. Mm-hmm. And do experiments, and that's the way we gain a lot of information by manipulating DNA.
Okay, so CRISPR, you take something out, and then does this protein still function? No, it doesn't. Okay, yeah. So then we know that segment that we took out, that codes for that
single. So you actually go in and manipulate one DNA letter, and that turns into one amino acid change in that protein. So that means that this specific amino acid in this protein is really important for that protein to function. And the technique is sort of, it's not super difficult to. It's difficult to use, but you can use it. you can use it with whatever DNA you have. That means that you can also do it on human DNA. So you can manipulate human DNA. And there was actually a Chinese scientist who did that some years ago. So what he did was that he made embryos that were resistant to HIV, the virus, and then he had two genetically modified human beings born. So actually, later it turned out that there was 3 babies born genetically modified, and that's no way to go. So we are not at that stage yet. So the problem is that using CRISPR.
What is the status of these 3 individuals? What is their health status?
So far, I think they are okay, but they're still young. So they're 2 years old, 1 or 2 years now. So what he did was basically just make them resistant to HIV, which it shouldn't affect their well-being. But the problem is that this CRISPR is not flawless. So even though you aim to only modify one DNA letter, you're not certain that it doesn't modify elsewhere in this. So we have this billions of these DNA letters, and the problem is that this method also can accidentally modify other DNA regions unintentionally. So it can make mistakes otherwhere, and that can be in proteins, for instance, that are really important for protecting us against diseases or against cancers and so on. So,
but we're bringing in like a hugely ethical topic right here. So if you can prevent a disease, do you do it?
Yeah, that's
the wording of that question. It was maybe unfair.
No, I thought you made it a little bit there. No, but it's important to discuss these ethical aspects of DNA manipulation. So the tools are there, but we just have to keep in mind that they also make mistakes, and more studies are popping up showing that they actually make more mistakes than we actually expect. So there are.
But just like the ability to read the human genome has improved in remarkably in 30 years, so too will the ability to manipulate DNA.
Yeah, I think
so. Take away the genes that might cause some diseases, and also to add genes for specific traits. So, for example, intelligence or athletic ability.
Yeah, no, but that's this ethical aspect, right? Should we make these designer babies that are smarter than us and look better than us? So there's a whole lot of ethical to this DNA manipulation, especially with respect to human beings. It's a little bit different with plants and lab animals. So there, for lab animals, it's done every day that we do these manipulations, but that's more to learn about how nature works, and not to set a new species wild in the nature that are gonna outcompete the ones already living in the wild. So that's in the lab. It's only to learn about nature. But when we do genetically modified human beings, that's a whole another story. And with the problems that these techniques still have inbuilt them within them, then it's there's still issues. So, but I still think we can. So there's an idea in using stem cells that we talked about in the beginning for organ replenishment. If we lost an organ, then you can ideally grow a new organ in a petri dish in the lab, and then put it back in this human being, or also for what do I know, neurological diseases like Alzheimer's, where you lost neurons and so on.
So to me, they sound like good things.
Yeah, no, but that's not improving the human being. It's more restoring what was lost.
But they also sound like good things that are only available to those who live in a country where the healthcare might provide them, or they have enough personal wealth to pay for them. So it kind of brings in a bit of elitism there to genetic modifications.
Exactly.
As would say the designer babies issue.
Yeah. But it probably goes as it did for the DNA sequencing right now. That in the beginning it was extremely expensive, but as these tools become more and more often used, the price also falls. Yeah. So, but this field is moving so incredibly fast that you have no idea. So I started in studying in 1990, and there it was. Yeah, there wasn't any DNA sequencing to the extent that we have now, and especially for DNA sequencing, it's been moving extremely fast.
So 1990 was that when the Human Genome Project started?
I don't recall the exact date when it started, but it was going on for several years to get this one human genome that costs many billions of dollars, and now you do it on an everyday basis to these sequencing. So I think that took more or less 10 years to get from this super expensive stage to this.
So whenever they're trying to, so the Human Genome Project, they were trying to read the entire instructions of all the DNA in. Did they have a range of different humans, or did they just choose one, or what? Do you know off the top of your head?
Yeah, no, actually I don't. But but for it to make sense, I guess it should have to be the same DNA, because we all got. So our DNA is very similar, but there are still small variations between humans. And for it to be complete, I guess they should make. Either they should take from one person and make that sort of the golden standard, or it probably make even more sense to take from several people sequence and then sort of take the average of those. So they sort out these few variations that we each individually have.
Yeah, but back then, if you're talking like billions and billions to do to do one, and then you got to make it viable, so you needed to bring in a few more subjects.
Exactly. To be honest, I don't know the exact setup for that one.
But since then, the cost of reading an entire individual's genome has dropped significantly.
Tremendously, yeah.
Do you know anything about these ancestry tests?
Not except the ones that. So there's a few persons in my former lab who sent DNA for that, and just got results back. So I think what they do is what they look at what's called SNPs. So that's small variations in certain number of. SNPs, that's it's called a single nucleotide polymorphism. So that's a variation in one single DNA letter, and that varies.
So instead of going through the entire, I don't know, billions of letters in a DNA code, they're just looking at segments.
They're looking at segments that they know that vary a lot between people from different regions of the world, or some are making you disease prone if you have a change in one single nucleotide. It's called one letter. If you have one mutation there, you're actually more susceptible, for instance, for diabetes. So there's a lot of effort going on today to map these SNPs to see to find out which one are making you prone to a number of diseases.
Yeah.
And that, of course, you can use to do this genetic testing for that. Of course, insurance companies like to, so they can decide if they want to insure you.
Are insurance companies asking for that?
No, but I don't think at the moment.
But if like I did do a genetic test and I went to a company and I sent it away, but then after I sent it away, I started to worry. Like, did I just send away, not just my social media data, but my actual internal data that a insurance company could, does this guy have any inheritable diseases? Or
of course there's whole ethical aspects of looking into your DNA. That's this because when you do submit to these ancestry tests, so they have this sort of basic package where you just sort of get your ancestry, where you come from and who where your DNA originates from, or where you originate from. But then you can, on top of that, you can also buy a sort of a health package, so they can screen you for if you're prone to a variety of diseases. And of course, if insurance companies start to request that, if you want to insure people,
that's a very bad way to go down there.
But
that's a very bad way to go down.
Yeah.
It's also, since I did send the test, I was also kind of doubting whether how it works, because the reliability is only as good as the amount of people that have sent in their DNA.
Yeah.
And if you follow like human evolution, we've all had a shared like journey from Africa to Asia to Europe, whereas mine didn't go further than Ireland.
Yeah.
A little bit Finnish, but that's about it. And I'm certain that somewhere down the line, I have ancestors from other places.
Yeah. No, but I think that the further they're able to go back, so of course but as you said, we already originate from the same pool of people in Africa. And I guess if you want to track further back in time, you have to do DNA sequencing of these corpses that are found, from people living back in several thousand years in Egypt and so on, further along back in time. Like for instance, Eske Willerslev is doing in Copenhagen, this famous DNA professor.
Does he wants to bring back a woolly mammoth?
I don't know if he's...
I saw a headline there someday, and you can't trust the headlines.
No. But he would have the knowledge to do it, or at least he probably have mammoth DNA in his bank of DNA.
Yeah, and the mammoth DNA wouldn't have degraded in the several thousand years since it was.
No, so the advantage, the advantage it is that for DNA that it is stored in ice, because it get degraded at what you would say room temperature, or this normal temperature around what we have here. But actually, so people are starting to look
just like in that documentary Jurassic Park.
Yeah. But but people are starting to, or scientists are starting to use proteins to sort of go back to the DNA code, because some proteins are really stable, and they are stable for millions of years. And if you can use another tool instead of DNA sequencing. It's called mass spectrometry, and there you sort of outline the sequence or how these proteins are assembled. So proteins are assembled from amino acids, it's called. So you have these 20 different building blocks that are assembled into proteins, and some of these proteins are super stable and stable for millions of years. So if you get the protein sequence from these million years old remains, then you can actually go back and
if you get the protein sequence, then you have the amino acid sequence, and if you have the amino acid sequence, you have the DNA sequence.
Yeah, because it has been outlined how. So DNA sequence comes in when you have a gene. It comes in sections of 3, so that's called codons. So and each 3 letter of DNA, that's translated into 1 amino acid. So if you have this 1 amino acid, you can actually go back in this table of the DNA letters and figure out how the DNA sequence could be. So that's one way to go back and build a being that's even older than you have DNA for. So that's.
Would you like to see a woolly mammoth in the wild?
It could be fun, but so my point of view is that science should also have a sort of a medical aspect.
Uh-huh.
So, but of course it would be amazing to to see such a thing.
But going back further, dinosaurs are too long ago. There's no proteins that can survive that.
No,
we're not going to see any dinosaurs brought back.
No,
okay.
They went extinct 65 million years ago. So that's
okay. That's a long time.
Yeah.
Okay.
That's too far.
Well, let's just take the opportunity to go back to like we're talking about ancestry there. So if we go back, we'll find our ancestors come. We have a shared ancestry in an ape. Then we go back further still. it's a fish. We go back further still. There's complex microorganisms. We go back further still. There's less complex microorganisms, and then we're back to the start of the world.
Yeah.
4.5 billion years ago.
Yeah.
And Earth is a barren land. Raining asteroids are coming from above. There's hot magma oozing up from deep in Earth's crust, and there's a sea, there's an ocean.
Yeah.
Do you have any this is entirely speculative.
Yeah.
Nobody has the answer for this, I'm sure. But do you have any explanation or your best idea about where DNA, this code that codes for all biology, where that came from, how that started?
So there has been, of course, some studies on this, and as you're perfectly, as you say, there's no proven experiment that's how it was. But I think there's a general consensus that most people think that it started with what's called RNA. So an RNA, that's an intermediate between DNA and protein. So when you have to make a new protein, the DNA is translated into this intermediate called RNA, and then the RNA is further. The DNA is transcribed into RNA, and then the RNA is translated into protein. And there's a lot of evidence that points to that RNA was actually, there was this RNA world to begin with, that's where life originated, and then it at some point in time you got this more stable DNA, and that's where you then stored your recipes, and then RNA became an intermediate. So the advantage of RNA is that it actually has enzymatic activity. Some of these, you have a whole lot of variety of RNAs, but some of them have enzymatic activity. And
enzymatic activity,
enzymatic activity, so it can actually perform a function. So an enzyme in the body is normally a protein that exerts a function. It converts one molecule to another one, for example. And that's how the body works. So when we metabolize the food, it's a whole lot of enzymes that process what we eat into to chemical energy that our body can use. So that's enzymes doing this. And there's some evidence that point to that RNA also can perform this enzymatic activity. Also that it can self-replicate. So if you have RNA, it can actually make copies of itself. So some of the RNA can.
So the RNA itself can act like an enzyme to catalyze chemical reactions.
Yeah, and also and make copies of itself. So that's also really important, for instance, for life to begin that you can actually make identical copies. And there are some indications that in this what's called this primordial soup, as you just described, this terrible atmosphere. There was no oxygen. There was ammonia, and it was really hostile. Yeah, exactly. It was really hostile. It has been shown that in this, if you make sort of a similar environment in a closed tube in the lab, you actually can start building these RNA molecules. It should also be said that there has also a very famous experiment from '52, I think it was, that where they were boiling water and let in methane and ammonia and those gases, sulfide, and then put electrical shock to it, so this sort of mimic lightning. They could actually build what they showed was 5 amino acids, these building blocks for proteins. And later, recently, the tubes from back then has actually been kept. And today, with modern analysis, they could actually see that they managed to make 23 amino acids in this experiment. So sort of all the building blocks that are still needed are used.
How many amino acids are there?
So normally there are 20 amino acids, but 20. Then you have different versions of chemical, different versions. But the
23.
Yeah. So yeah, it's probably more. So, but there are a variety. But that experiment, they could make 23 amino acids.
So that's interesting because amino acids can actually form themselves without biology.
Yeah, based on that environment back then, they could do it. And in in this experiment where they were mimicking the situation back then, they could actually form this amino acid. So that's why so most people believe in the RNA world. And
Okay, so that's very interesting indeed, because the RNA has the instruction to build amino acids, but then you can have amino acids in the environment.
yeah.
Yeah.
And there's also another point that's really favors this RNA world is that this complex that translates the RNA into protein. It's called a ribosome. So the what's called the active site in this whole complex. So that's where the enzymatic function is going on. That's made of RNA only. So that's everything that goes on there is controlled by RNA. But so the whole ribosome unit today is a merger of protein and RNA. But where the stuff really happens, they have RNA. So that's favoring the RNA world hypothesis.
Yes. And then at some point after that, do you think that the first microorganisms could have lived with RNA in their cells instead of DNA?
Yeah, that's very likely.
That's the
but
the
it is
hypothesis.
but RNA is much less stable than DNA. But there are, for instance, there are viruses that have their genetic information stored as RNA, and then that get copied in whenever a virus infects a cell, it makes copies of itself and makes more RNA, and then the new virus particles are assembled with RNA as the genetic information.
Well, let's talk about viruses, because how far back do you think viruses go in our 4.5 billion year history on Earth?
I think they go pretty far back.
Yeah. So close to the beginning of biology.
Yeah. I don't know.
Of course, it's hard to say.
Yeah, it's possible that somebody has really done some, digging into it. But but how far back they go, I don't know. But there are some. I think there's called transposons within your DNA. So that's basically just small pieces of information that jumps around in your. So that's also sort of a.
So viruses, they do what everything else in biology does. They replicate. They replicate the code.
Yeah.
And they do it well.
Yeah. Some of them are really efficient, too efficient for our tastes.
Yeah.
Yeah. But so that's basically, so evolution is all about copying your DNA. So an evolution success. So evolution is success. You can say that's measured in one parameter, that how much DNA you got of a certain species. The more DNA you have, of course, that putting a little bit harsh, but the more DNA you have, the more evolution successful you have. So that's why humans
okay, I haven't heard that before actually. So the more DNA you have in
no, not the amount of DNA, also the number of copies.
The number of copies.
Yeah. So the more sort of live being of more copies of human beings, the more evolutionary successful you can say that we are. That's one way of.
The more babies we have.
Yeah, but but we are an evolutionary. We have an evolutionary success because we have we keep expanding in numbers and we sort of taking over the world.
Yeah, but could you not say that viruses are doing that better?
Yeah, but so you can also argue which one are most successful.
Yeah.
So some of them, I read a book called Homo sapiens where he argued that wheat was actually domesticating people, because so what we're doing now is we're really taking care of the wheat plants and make sure that they are planted all over the place.
Yeah.
And really nurture them.
And if it wasn't for us, there would be hardly any wheat plants. But we're cultivating wheat plants, so wheat plants have it all figured out.
Yeah. So they and
we're tools for the wheat plant.
Yeah.
But also if you look at the like our ancestors, from Like the archaea, the first bacteria, they're still around. You know,
Yeah.
they've changed a little bit. They've changed a little bit for sure, but they're still around. You know, they haven't changed a lot in billions of years.
Yeah. No, that you can also say as an evolutionary success that they have made something really really solid and
Exactly,
and
it
still
works.
around.
Yeah. No, there was no need for anything else.
No.
So, Richard Dawkins, 1976, the selfish gene theory, that where all biology is just mere vehicles to carry the code.
Yeah.
So, the life can be, you know, summed up as just a movement of the genetic code from one generation to the other.
Yeah. That
With
the
slight-
genes only think about themselves.
With a
so
gut.
that, yeah.
Exactly.
So yeah, so I read the book and it's an interesting way of looking at it, but I think it's more philosophic than anything really useful approach because but it's an interesting idea that the genes just make sure that they are copied and then they just have to make sure that they invite environment with other genes.
Well, in a human society, it's very complicated because we have culture, culture brought in that dictates what is right and wrong behaviour, dictates, you know, how we live. But I guess if you look at any other species, you can see, okay, they're in a way programmed to find food, find shelter, find a mate.
Yeah. So this. I know that Richard Dawkins really claimed that this altruism in amongst animal species it doesn't really exist. There's no true altruism. So what you do is that the one that really takes care of the species is the one or your ancestors directly above your sort of mom and dad or grandparents. That's in the animal world. That's really they are protecting their own offspring, but not anybody other from that species. So it's only your very own gene pool that you really want to make sure that it is preserved.
Yeah.
So
But then in a social society, if you help other people, they're going to help you. They're going to return the favor.
Yeah.
You know, and we're it's a mere fluke that we're living in cities right now. Didn't have time to biologically catch up on living in cities.
No. So that's also one debate now. If evolution is if we have put that to a halt, but the thing is that evolution really takes several thousand years to adapt to changes. So it's so we can look at the past few generations and say that we have sort of put evolution to a stop because it takes many years before we can really monitor and say anything about that. So evolution really takes generations of a species time, and so it takes many thousands of years before we adapt. So that's also why people believe there's so much obesity these days because we have what's called the thrifty response. So in some ways we are still cave people in genetically speaking. So that means that our body is set to it, but that when it sees food, it really has to preserve it because it's not certain that there's going to be anything tomorrow. And that's a problem today where there's surplus of food. Because whenever we eat, our body really makes sure that it stocks up and build these adipose stores so that we have this extra food.
Yeah, it also gives us a desire to have that that kebab.
Exactly. Even though we know it's not good for us, because our body really strives to build up extra energy because it's not so sure there's going to be anything tomorrow. So. And that's something that we inherited from back from the cavemen.
Whenever we had to work for our food.
Yeah, when food supplies were much less certain.
Yeah.
So that's a perfect example of how we have moved way faster than evolution.
Yeah. Can I get another beer?
Yeah. good.
Cheers.
Cheers.
And Philip, you're a scientist, an academic. You're an expert in DNA, but you've also wrote a couple of novels.
Yeah.
Fictional novels.
Yeah.
with a lot of scientific input.
Yeah.
Can you tell me about them? Your first one. When was that published?
So that was in 2016. And it was something that I've been working on for, I don't know, 4 or 5 years. So it sort of annoyed me that there was people really exploiting science when they were writing novels. So they were over-exaggerating what you can do with science. For instance, there's Dan Brown in Angels and Demons. He has this little container where he has antimatter in and you can't have that.
Yeah.
So I always wanted to write a book, and then I decided if I should use my knowledge, how bad can I really make it? So what I wrote I saw a new heaven and I saw a new heaven. It's called.
It's only in Danish at the moment.
So actually, it is we translated together with an Irish colleague.
Okay. Yeah.
So it's available on Amazon but there it's called Cortum.
Cortum.
Cortum. Yeah.
With a K.
C-O-R. Yeah.
C-O-R-U-M.
D-O-M. Yeah.
Okay, Cortum.
Yeah. So there, I really wanted to make the worst scientific experiment you could imagine, and that was, of course, killing people using science, and as many as possible. Of course, there's an angle to it, so it sort of has to make a little sense in the book.
But I don't want to give too much away, but a SARS virus did feature in the book.
Yeah.
4 years ago.
Yeah. Oh, but it is so it's based on. So there's a lot of real science in it. So I draw parallels between the virus that I made in the book, or came up with for the book, and then the real viruses like SARS, which was that. So that existed mainly in Hong Kong or East Asia in the start of this millennium, and I had a personal vendetta against that, because at that time my brother was in Australia and we were planning to go see him, but when then SARS took over and we heard of rumors that some planes were kept in quarantine in our Australian airport for 14 days, so we didn't dare to fly to Australia at that time.
Wow.
So, but that was my.
I haven't heard about that with the most recent SARS outbreak.
No.
Corona. Yeah.
No. But that's
so that motivated you to write the book, because.
Yeah. So I had this personal grudge with SARS. No. Also, I don't know. It was just. I just wrote that into the book just for my own sake. But,
okay.
Yeah. No. So the motivation. So whenever I was a kid, I've been reading many novels. So I always wanted to make one my own. So I just had to came up with a story, and that's sort of developed. It took some years at the end of my PhD and afterwards when I was a postdoc, and then I started writing in, what was it, 2011 or 10, I think it was, and then sort of in the end it took a long time, but then you sort of get the feeling for it, and then you can speed up.
Well, you did, because you came out with a second book.
Yeah. No, but things are moving faster now. When sort of you learn how to do it, and also how I make myself most efficient. You know, I need to have a table where I say, this week I have to write this number of words and so on. So I have a sort of a schedule to push myself. But still, it's The second one is also about science. It's actually about evolution.
Well, I enjoyed a quote about the book. Fenris is a story about evolution and what can happen when people misunderstand the most fundamental law of nature.
Yeah. Oh no, yeah, because so the scary thing is that there's a movement, and that's mainly in the U.S. that evolution is not true. That there was this divine thing that created life on Earth. So and that's sort of, I don't know if it's scary, but it's they're teaching this in school that kids don't really learn about evolution anymore, and I think that's scary because it's a fundamental aspect of nature that so it's simple to understand even for kids that the most or the best adapted one will actually be the one succeeding in making copies of itself or proliferating. And now that you suddenly teach in school, kids teach in school that's not true anymore. That's so, but there's a whole movement. I think that's a whole anti-science movement.
Yeah, but it also that kind of teaching separates man and nature.
Sorry?
That teaching separates man and nature.
Yeah.
You know, in the heads of those who hold the idea.
Yeah.
So then, you know, their whole behaviour throughout life is going to be to assault and attack nature, and
yeah.
That's not a good thing for the rest of us. So beliefs like that and ideas like that, it does affect the rest of the world, I think. So it's good to try and try and do something against that. And what I also think is that whenever people who read science are going to, you know, follow the ideas and have a good sense what, for example, evolution is, but people who don't read science are not going to get into that.
Yeah.
Pop culture is the only way to really reach a lot of people.
Yeah, I certainly hope so.
Yeah.
That this, because it is important that people have a grasp of science. Mm. Of course, they shouldn't be expert or scientists themselves, all of them, but it's still important to understand the nature around us and how it works to a certain extent.
Can you give us a little teaser for Fenris?
Yeah, so Fenris is so there's a story in the past and then the present. So in the past there was this wolf, or it's not a wolf, it's a dog that turns into very similar to a wolf. So that's where the title comes from. That's his Fenris. And that really relates to what's going on in the past. So in the past, you follow 3 different characters. There's a girl, Asta, who lost her mother and is really trying to get back on her feet. And then as the book goes on, she discovers more and more about her mother, which was a scientist actually at the University of Southern Denmark.
Nice.
And but has to that one is actually local to Odense. And then you have another young girl called Cindy, which has a child. She's really a severely handicapped child, and she's really struggling to live this single mother life with him being really handicapped. But then he suddenly at one point shows a novel side of himself that she didn't expect. And then you have this grown-up man in the end that he's working at the hospital and he has he has an old debt to pay that relates back to Fenris. And as the story goes on, these 3 different people, their stories merge into one. And then Fenris come into play and it sort of explains why they merge into one. Mm-hmm. So and that's
gotta read the book.
Yeah, you have to. Yeah, write to me and buy it.
That's available in Danish. Is it going to be in English as well, perhaps somewhere down the line?
So this colleague of mine, she moved back to Ireland. So probably not at the moment. So it took a long time to translate it. So we did it. So I did the first translation, rough translation, then she, perfected it. Yeah.
Well, I will link to both of those books in the text of this podcast for anybody listening.
Great, thanks. But it's it's something I can't stop doing it. So I've continued and I'm writing on a follow-up for the first book. So with the same main character called Martin. And he's on to a novel quest So yeah, he's and that goes sort of it draws. It also goes back in his history and have involvement of the Hitler's Nazi.
Yes.
Germany and so on. So that's always a good thing to put in a book. If it's something really the meanest people who ever walked this earth.
well, yeah. So they were also they were proponents of genetics for all the wrong reasons.
Yeah. They were so But that's yeah. So I'm writing on now, and hopefully it's gonna be out. Not next year, but the year after. I hope. For all of them and all the comic books, there's definitely gonna be a science part of it. That's the most important thing.
And actual science.
Yeah. When you work in science, you have to sort of lure the secrets of the nature. That means that you do a lot of mistakes. So you do a lot of guessing and then develop theories, and then they can turn out to be wrong. When you write books, your theory is always right, but you don't. So you don't make. So you're the one deciding how the story goes. But whatever science or plot I put into it, based on science, it has to be real science. It has to be doable, sort of say. So that's also why I end the books with a chapter where I comment on the science that I put in the book.
Philip, how did you get into this world of science? What was your motivation?
So so when I was a kid, I was always interested in nature, and I recall that for my confirmation, or this when I confirmed my belief in God as a youngster. So when everybody else wanted stereos as presents from their parents, I wanted this animal encyclopedia from a Danish publisher. So I got this. So it's a whole range of book about the animal kingdom. So I've always been interested in nature and animals, especially animals. And then in high school, I had a teacher called he was called Bjarne Poulsen, and he was he was untraditional teacher, but he was very provocative and really taught us how to think in a different way, and he really got me interested in science and especially DNA and really the molecular biology of science. Mm-hmm. So I think that it's very much Bjarne's thanks to him that I'm here today.
So as a kid, you're interested in the diversity of the animal kingdom, and then you go to high school and Bjarne teaches you that it's DNA that codes for all of that diversity.
And at that point, what of course, mingling with DNA at that point was very basic, but I really like this concept of DNA and how important it is and what you can do with it.
Yeah, it's a fantastic story.
Yeah, but it began in childhood and then really exploded during high school.
Just honest curiosity.
Yeah,
that's a good way to.
That's the most important thing when doing science. That's this curiosity.
Indeed. Well, Philip, thank you very much for joining me.
Thanks for having me. It's been a true pleasure.
Cheers.
Cheers.
Hope you enjoyed that episode of the Science and Beers podcast. If you want to keep track of Philip's work, he's got a website, philiphallenborg.dk, and I will link to his 3 book publications in the description. Also, please consider supporting the Science and Beers podcast. You can do that at patreon.com/scienceandbeers. Thank you.